Okay, so we're on adaptive immune response, and our next objective is to be able to diagram an antibody and label these important sites. So in your book, they've got some diagrams, and they've made some improvements over the years. I don't know if it's whether some of the complaints that I had sent them or what, but antibodies are also called immunoglobulins because they're a product of your immune system. They are proteins. They are somewhat globular in structure. These simple Y drawings that we do are almost like stick figures we do for people, right? Not the level of detail that the molecule is in real life. So this overall characteristic Y shape has two general parts. When we break it up, when scientists were studying it, they could break it into two fractions. That's what the F stands for. This top fraction, and actually it would break into these two individual pieces, sometimes referred to as the arms of the antibody. And this portion down here, commonly referred to as the stem. The stem is the FC fraction, and C actually stands for crystallize. This actually crystallized when they were doing their experiments. These two portions, the FAB, stand for antigen binding, because these are the parts that bound to the antigens, these regions of the antibody. So these are very important functionally for how antibodies help us. They specifically bind to antigens. At these top two sites are the antigen binding sites, these little curves that you see indicated here. But when you go into a little bit more detail, like these two drawings do, they're actually made of two identical protein segments that you have two copies of. You have this shorter segment here that's on both sides of the antibody. These are referred to as the light chains, as opposed to the longer segments you see here in the middle, two identical ones, referred to as the heavy chains. Because, of course, the light chains are smaller, so therefore they were lighter, and the heavy chains are longer, so therefore they were heavier when they separated out these four different proteins, when they were able to break it into the four pieces this way. So as you can see, the light chains are attached here to the heavy chains, and the two heavy chains are attached to each other. So you get this overall characteristic Y structure that's very simplistically written here. But when we get into the actual structure of each one of the chains, there are two distinct regions that each of the chains have. They both have a variable region, and this is associated with the antigen binding site. So this what makes each antibody different from each other. Your body produces millions upon billions of different antibodies, each one of them binding to a different antigen. But on the antibody itself, both of these sites are the same. The heavy chain segment is the same for both sides, as well as the light chain variable region. So they could basically grab onto two antigens or two sites on the same antigen at the same time. The bottom portion of the heavy chain and the bottom portion of the light chain is referred to as the constant region. So from antibody to antibody, this is pretty consistent. And the reason for that is it has to be in order for here, for the heavy chain and the light chains to bind with each other, right? If you didn't have the same proteins here, this binding wouldn't happen. And so you wouldn't have heavy chain and light chain together, and you wouldn't have this variability of the antigen binding site that happens by having both heavy and light chains binding with each other, these two protein segments. Now, this portion, the FC region, right, the constant region, as well as the heavy chains, this portion of what we call the stem, does vary between the 5 different classes of antibodies, right? But within a class, it's pretty consistent. And again, that's so that our body can recognize it. But we actually have 5 different types of antibodies that your body produces. And this portion, this stem portion, the FC region, is what determines which one of the 5 classes that antibody is. And that will also determine how the body responds to the antibody. Because this is the portion of the antibody, the stem, the FC region, is what your immune cells interact with and components of your immune response interact with. The top portion, as the name implies, right, is for antigen binding. So this is a relatively simplistic, right, again, kind of stick figure of the antibody. And you can see here that orientation, right, of it binding to, say, a bacteria. The antigen binding sites, the arms, and the stem, the FC region, is sticking out so that the immune system can interact with it. So let's see. I'm going to go over to my other PowerPoint that I'll post for you guys. So hypothetically on the test, right, I could label each one of these, right, a different letter, and you should be able to tell me, right, multiple choice wise or matching wise, what each one of these letters represent, right? So for instance, for A, that's the variable region, right, of the heavier light chain, right? It's the shorter chain. It's also, right, associated with the antigen binding site on this side. This E represents the constant region of the light chain. What do A and B represent here? Antigen binding sites. What does C represent? The constant region of the heavy chain, or the FC region, or what I'm trying to show you guys here is this is where the immune system would interact with the antibody, right? With this portion, this FC region, the constant region of the 2 heavy chains. All right, so these are the important polar ends of the antibody, right? This is where it's binding to antigens. This is where it's interacting with the immune system. So, of course, heavy chains are in green here, right? They're the center. And then on either side, mirror image, are the 2 light chains. And it's this combination of these 2 proteins that gives us the variability that we get at the antigen binding site between the variability of your light chains and your heavy chains. So remember, we can break it up into 3 pieces, right? Your A's and B's is your FAB regions, and this stands for what? What does the A and the B stand for? Antigen bindings. Remember, these are the ones that have the antigen binding sites, these 2 fractions. The C fraction, right, crystallized, that's why it's called FC, but this is, again, the component of the antibody that interacts with the immune system. So this diagram, what am I showing you here? What do you think? What am I trying to emphasize here? What regions? What do you think the greens are representing? It's on both the light and heavy chain. I've got it highlighted, right? And it's this near where the antigens bind. So this is the green is your variable regions, right? This varies from antibody to antibody. Where the blue represents what? The constant region for both your light and heavy chains. And this, as you can see, denotes the structural integrity of holding all of this together. Light chains binding to heavy chains binding to each other. So there's a reason why you see this organized in the way that it is and how it works. All right, so its structure, what's where, is very important to the functionality of these antibodies.
What does the C stand for again? I know the AB is antigen binding. What does the C stand for?
It actually stands for crystalline. It crystallized. But the important thing to know is that this is the portion, right? The constant region, this region, of the heavy chain, the stem, or the FC region, I always refer to it as the FC region, is what your immune system actually interacts with. So those phagocytic cells, right, when antibodies bind and a phagocytic cell comes along, they have a receptor for the FC portion of certain antibodies. And so they can bind to this section, and then they'll, remember the first part of phagocytosis is attachment and recognition, right? So it's recognizing it by the antibody attached to it and attaching to that antibody, which is attached to the antigen so that they can engulf it. And that process, when you're tagging it for destruction, is called opsonization. So antibodies can opsonize, tag for destruction. What other protein that we talked about in the complement system, it has a letter and a number designation to it? C3b, right? Remember, B for binding. Binds to the surface of those cells and acts as an opsonant for our phagocytic cells to grab onto and recognize that pathogen and engulf it. So we have two very strong opsonants. C3b isn't very specific, right? It's a product of our innate immune response, our generalized response. Antibodies, on the other hand, are very specific, and they're a product of our adaptive immune response. And bacteria could get coated in C3b and antibodies, right? It's double tagged for destruction, right? It'll really get destroyed. We have two signals screaming out, destroy this thing. So again, right, I wrote out here, right, the FAB regions include all these letters, right, A, B, C, D, E, F, and G and H, right? This is all that antigen binding fraction. Your FC region is this bottom portion, right, of the heavy chain, this constant region, I, J, K, and L. The constant regions, remember, extend a little bit further, right? The bottom half of your light chains and just past your variable region for the heavy chains. All of this portion is constant. Variable is just up here at the top portion of each of the light chains and heavy chains is what varies from antibody to antibody. But in truth, the light chains, these two are identical to each other, right? The heavy chains are identical to each other. But it's the combination that we'll talk about later of the sequence that's here in this part of the protein and the combination that's different here in the light chain that gives each antibody its unique antigen binding sites, represented here by M and N. And then again, O is just representing and reminding you that this is where the antibodies interact with the immune cells and components of our immune system. Because you guys remember complement again, right? It's part of our innate immune response. One of the pathways, the classical pathway, can be turned on, activated by antibodies. The protein from complement binds to, guess what? This region of the antibody. So these two things work together, right? Even though we talk about innate and adaptive kind of separate, right? But they overlap with each other and they help each other out. The difference is that your innate is very generalized, right? And it does not change over your lifetime. Your adaptive, on the other hand, is very specific and it adapts and changes over your entire lifetime. And then one very important component of your adaptive immune response is, remember, when we activate B cells that make antibodies and T cells that help with that and cytotoxic T cells, what other set of cells do we also make? Memory cells, right? So we remember this. We remember the specificity, right? Some of these antibodies, too, stay around for a very long time in your body to help protect you after you make them, whether you've been exposed naturally or artificially by vaccination. All right, so end that one. Go back to and some of you guys may have noticed I skipped over, right? We didn't go over nature of antigens, right? I bet someone was going to call me out on that pretty soon. There was a reason why I had done that, but I am going to come back to it. All right, so these are all the wonderful things that antibodies can do, right? So when an antibody binds to an antigen, how could that help us, right? And let me see if I didn't number these. Okay, good. So I can go in any order I want. All right, so the one we've already experienced is opsonization, right? When antibodies bind, phagocytic cells can grab on to the FC region, right, the stem portion, with receptors that they have. They're just not showing you here in this picture, and they can engulf it. Now, what's a phagocytic cell? Specifically a white blood cell that has the ability to do phagocytosis. Macrophage, neutrophils, dendritic cells, right? Basophils to a smaller extent. But your main phagocytic cells, right, are your macrophages, your dendritic cells, which remember can go one step further and present information that we're going to talk about some more later, right? And your neutrophils, right, which are only going to go into your tissue during inflammation, though, right, when they're called there. Your dendritic cells and your macrophages, though, are always there, right? So when antibodies coat them, right, this is going to help signal the phagocytic cells to destroy them, tag them for destruction, opsonize them. And as we talked about before, right, the complement protein, a particular complement protein, can bind, and again, notice where it's binding to that stem portion, because the antigen binding arms are grabbing onto the bacteria. And this will activate complement. And remember, the major outcomes of complement is, one, you're going to produce C3b, right, which can bind more and opsonize even further. You're going to get an inflammatory response. And there are two complement proteins that you need to know that are directly related to inflammation. Do you guys remember what they are? C3a and C5a. And then lysis of foreign cells, what causes that? What's the name of that group of proteins that inserts into the membranes? It's not a Big Mac. It's a Mac complex. What does that stand for? Membrane, right? That part is important, right? Because it only inserts into membranes, not cell walls. Membranes. Membrane attack complex. So all of these things can happen when complement is activated. One of the ways complement can be activated is by antibodies. Do you guys remember the other two ways in which complement can be activated? The alternative pathway, what has to happen? C3b binds. The lectin binding pathway, right? We have receptors that are recognizing complement proteins that recognize that mannose sugar, mannan, associated with microbial membranes. So there's three different ways we can activate complement, but you're always going to get, right, the possibility of these three outcomes, right? Once that cascade is activated, you're going to get more C3b produced for opsonization, tagging for destruction. You're going to get an inflammatory response, two proteins important for that, C3a and C5a. And you're going to get the formation of the MAC complex, the membrane attack complex, which can lyse foreign cells. All of those things could happen. The next type of protective outcome that you can get from antibodies is immobilization and prevention of adherence. So especially bacteria, say, that are motile by flagella. If we have antibodies specific against their flagella, remember their flagella rotate like a propeller does on a boat? So if you have antibodies attaching to them, it's going to kind of clog them up, right? So imagine, and this actually happened to me, putting down the bayou, right, on the boat, and all of a sudden the boat stops. We're like, what the heck is going on, right? We pull up the motor, there's a sweatshirt wrapped around the propeller, right? So we had to take the sweatshirt off, right, to be able to free up the propeller so we can keep going down the bayou. These bacteria, though, when the antibodies bind to their basically propeller, right, their flagella, it's going to clog it up and immobilize them. They're not going to be able to move, right? They can't pull the antibodies off their flagella. So that helps in immobilizing them. And the very first thing important for all infection, right, for us, is they've got to be able to attach to us, right? If they can't attach, then they can't stay, right? Things like our mucus will wash them away. They can't stay put unless they attach. So lots of bacteria like gonorrhea have specialized fimbriae that we talked about, those papillae that are specifically for attachment, right? The bad news with that one is it's always changing though, so we can't make antibodies against it. If we did, we could attach to those fimbriae and stop them from attaching to us, right? Stop that very first crucial step of infection, of attachment. So we can prevent adherence, prevent them from attaching to us by creating a barrier, by having that antibody bind to them first so they can't bind to us, right? We create that barrier between the two. And this is very effective, too, for even viruses, right? They've got to attach first. If we have an antibody that binds to them first, then they can't attach to us. When antibodies bind to viruses and toxins and basically stop their specific attachment to our cells, we refer to that as neutralization, right? Because we've essentially stopped them from being infectious or damaging. We can neutralize them. As we saw when we were looking at the structure of the antibody, as I said, it has two antigen binding sites. They're both identical. So one single antibody can bind to two different antigens at the same time, in this case, two different bacteria. When that happens, you can get these aggregates, this cross-linking that happens. And there's two types of cross-linking that can happen. Agglutination is when you have very large things like bacteria, or in the case of when you're doing blood typing, right? How many of you guys have taken AMP2 lab, right? When you're doing blood typing, you're sticking red blood cells in with what? Antibodies. Antibodies, you know what they bind to, right? You have an antibody against the A antigen, an antibody against the B antigen. When you combine that with red blood cells, if those antigens are there, the antibodies bind to them, and you get that clumping, that agglutination effect that you can see with the naked eye, right? You don't need a microscope or anything because the red blood cells are so relatively large. You can see this even with bacteria, right? If you have a large amount, you'll see this agglutination that'll happen. Then, of course, looking at it under the microscope will even help you visualize it even better. Other times, you have very small things like proteins, right? Bacteria, red blood cells, these are very large. Very small soluble antigens like proteins. In solution, you wouldn't see them. But when antibodies bind to them, it makes it a very big molecular complex. An antigen-antibody, or what we call immune complexes. These will settle out of a liquid media, right? You'll see precipitation. Right? That's what that means. A lot of times they'll run these types of reactions in an agarose gel, right? So it's kind of a gelatinous type media, and they'll put antigen on one side and antibody in the other, and they'll diffuse towards each other. And you'll literally see a visible line in the agar where the antigen and antibodies are binding with each other, right? So because you have so many molecules, you can see it with the naked eye. It's such a large complex. Now that's outside of our body running tests, right? What benefit would clumping them up in our body be? Why would you want to clump them all up in one little area? Yeah, why? Why would it be beneficial? Right, to prevent it from going elsewhere. If we can keep them trapped in one particular area, right, it's going to be easier for your phagocytic cells to engulf them and get rid of them. The problem is that when these complexes persist or get into the bloodstream, and that's one of the disorders we'll talk about later, where immune complexes, antigen and antibodies build up, right, and don't get cleared, right, and this can create blockages and problems, especially when it happens like in your kidneys, right, where filtration of your blood happens. It can really interfere and cause damages there. Make sense? Okay. So the very last one stars our natural killer cells, right? That one subset of lymphocytes that's not antigen specific, but has some pretty cool abilities in killing cells. One of its abilities is via the use of antibodies, and only one class of antibody actually has this ability to communicate with the natural killer. So you'll notice that I have IG written here. That stands for immunoglobulin. And then the second G stands for the class of antibody, and that's the G class. Each one of the classes has a letter designation that we'll go over in a moment. So it's only IgG, this one class of antibody, that can do this one special ability with the help of natural killer cells. It's called antibody-dependent cellular cytotoxicity. So it needs antibodies. The antibodies are what are recognizing the cell, right, because this is cellular, that is maybe infected by a virus or a bacteria, and it might be presenting those proteins on the surface, and we've made antibodies against it. So those antibodies are binding to the cell. The natural killer cell comes along, sees this interaction, can bind to the FC portion, right, of this class of antibody, and that's going to signal this cell to release cytokines and toxic substances to cause the death of this cell, right? Because clearly there's something wrong with it. We're specifically identifying something's wrong with a specific antibody. Mm-hmm.
So that's what I'm giving.
They're giving you antibodies. When they give you an immunoglobulin, they're giving you antibodies. It's another name for an antibody. And it's probably specific against a particular infection. What did you have to get immunoglobulin for?
Well, I have ICP,
Oh, wow.
no.
So it sounds like probably you don't produce
my
your...
spleen is strong as my plate.
Right.
So
you got to have... Why are they giving you antibodies, though? I'm surprised I don't take away your spleen. I don't know that specific disease, but usually when they give somebody antibodies, it's because you're not producing enough antibodies to help protect you. And there's lots of different disorders out
I
there.
only had to get it twice through infusion.
Mm-hmm.
That's when my platelet count got real good.
Yeah.
3,000.
Yeah.
They did give me the option of having my spleen removed, but I haven't had any complications.
Right.
My
So
platelet count is at a good level.
right. So don't mess with something that ain't broke. Yeah. Don't cut into me if you don't absolutely have to, right? So if you have a serious flare-up, then, it'd be something to consider. Yeah. So, yeah, immunoglobulins have a lot of great benefits to them. And so that was probably one of those things that helped give you some type of protection while you were in that weakened state where you don't have as many platelets as you should, and that help protect you. So notice, too, that, as I said, it's the classical pathway that can be activated of complement. But again, within that, only 2 of our 5 classes of antibodies can actually interact with complement proteins. And that's IgM and IgG, right? So that's one thing we'll go over again, right? But some of these antibodies, right, are, you could say, specialists with different components of our immune system and have different abilities. So as we've always talked about with all our protections, right, this is how it's supposed to interact, right? The antigen binding sites are supposed to bind to the pathogen. The FC portion is supposed to be sticking out so our immune system can interact with it. Well, there are some pathogens out there, and I think Streptococcus prurigens, the causative agent of strep throat, is one of the ones that's developed this ability of being able to grab onto our antibodies by their FC portion. So they can coat themselves in our antibodies in the wrong orientation. So they're grabbing onto the FC portion, the antigen binding site is sticking out, and so our immune cells can't recognize and can't get to these guys. Right? They're using our own proteins, our own antibodies against us. So for every defense we have, there's a microbe out there that's found a way around it. This is why we still get sick. So these are our 5 classes. And most of them are always in this monomer form, this very simple Y structure that we've looked at, where you have the 2 antigen binding sites, right? Your heavy and light chains, that stem, that FC region. On your B cells, the actual B cell receptor is actually the IgM class of antibody in this form. And so the FC portion is just embedded into the membrane. And then the antigen binding sites are sticking out to interact with antigen. IgG is always in this monomer form. IgA, when it's in your serum, in your blood, in the liquid phase of your blood, it is this monomer form. But most of the time we make this to secrete it into our secretions, right? So what are some examples of our secretions? Sweat, tears is a big one that we produce a lot of IgA in. Saliva, mucus, right? That's the big one, right? We produce gazillions amounts of mucus each day, and even more so when we're sick, like right for me right now. So in addition, for women, they produce it in their breast milk. And so this gives protection to the baby, helps line their mucus coating their intestinal tract. So this is one of the main reasons too why people nowadays more and more are trying to breastfeed instead of giving formula, because you actually give a protection that we can't replicate in formula, right? Not inexpensively anyways. So you're giving your child this level of protection when you're breastfeeding. You're giving them actual antibodies to help coat their digestive tract. And they've done studies, of course, right? And children do better that are breastfed as opposed to formula fed. So when it's secreted, though, it's in this dimer form. And so notice the FC regions are attached to each other. But remember, secreted means it's actually leaving your body. Even when we're talking about our intestines, that's actually an external environment to our body, right? It's a tube that goes right straight through. So there's no need to interact with phagocytic cells or anything else, that FC portion. What type of protective outcome do you think this antibody is doing in our secretions? So let me rewind. Which one of these protective outcomes do you think, or which ones of these protective outcomes do you think IgA is specializing in? Immobilization, right? So if it's a motile bacteria and it's in our mucus and we put antibodies on it and wrap it up, it's going to not be motile anymore, right? So that's really going to help our mucus push it away. Awesome. That is one of them. What do you guys think? In this same block, what can antibody binding do? Stop it from binding. Stop it from adhering. So for viruses and toxins, we could basically neutralize them, right? One more. Cross-linking. Again, clump them up, right? So that they're all in one particular area, easy to remove them. You can't get opsonization because this would need our white blood cells, right? And these aren't outside of our body. Complement is not outside of our body. Natural killer cells aren't outside of our body. So we couldn't get these 3 protective outcomes, but the other ones we could. Right? So that's why, too, last time I went to my son's doctor, he was like, am I going to get the up-the-nose medicine? and the doctor's like, the nurse actually said it hadn't come in yet. So there's 2 different types of the flu vaccine. You can get the shot, or you can get the nasal spray. And I highly recommend you do the nasal spray. Because how does the virus enter your body? Not in a shot. Up your nose, right? You inhale it. So getting vaccinated that way is going to stimulate your body to produce more of the IgAs. So then you have the IgA antibodies in your mucus. So hopefully you can neutralize that virus in your mucus before it even can attach to your cells. Right? So it's not necessarily for those of us that don't like getting shots. Right? My son actually would prefer the shot than the up-the-nose thing. He does not like the up-the-nose thing.
It's not a mist.
It's like a mist, but he's sensitive. And so I highly recommend, and even my doctor's office, because of the different type of immunity you develop from the nasal spray, go with the nasal spray as opposed to the shot, right, if given the choice. So IgD is one we don't know much about. It's always in this monomer form. IgE, I always refer to this one as the one Erica hates. This is the one typically responsible for allergic reactions. Right, so this is the one, the class of antibody that you produce that gives you the particular response that we commonly refer to as allergy. Because, again, what cells and what processes it activates within your body. So recently in some of the literature I was reading too, IgD is also found on the surface of the B cell. That particular class. IgM, when we secrete it, it is secreted from the plasma cells, the B cells that actually secrete antibodies, in this monomer form. So I always remember M for mega big. Not monomer, excuse me, pentamer. There are 5 of them hooked together. So as you can imagine, when you look at this structure, again, what type of, and these guys pretty much stay in your blood. What type of help is it giving you? Neutralization, yep, definitely would be good neutralizer. Which one do you think it would be super good at? Because, I mean, look at all those antigen binding sites. 10 antigen binding sites. Nope, remember natural killers, only IgG. The cross-linking, right? These are really good in agglutination. And this is the one you produce against the ABO antigens, or the AB antigens in the ABO blood group. That's why they agglutinate really well. That's also why you don't have to worry about matching your baby as far as ABO antigens go. Because this antibody is so huge, it doesn't cross the placenta. Not true, though, for all antibodies. What am I doing on time? So IgM, right, the first antibody that you produce, you actually can produce this as a fetus. It's the only antibody class the fetus makes. And again, we don't have to worry about mom, it affecting her. It doesn't cross the placenta, so it stays in the baby. Right. It makes up about 5 to 13% of the antibodies in your circulation. Again, stays in your blood, though. Too big to get out. And again, it's found on the surface of your B cells. This is your B cell receptor. So this is number 1 on your list, right? IgM. It's on the surface of your B cells. And it's going to be the first one you make, even as a fetus. And when you secrete it, it's going to be this huge, mega big pentamer. Sometimes T cells aren't needed in the activation of B cells. So the type of antigens that can elicit antibody response without T cell help, right? So T-independent antigens. So T-independent means we don't need T cells for certain antigens to get the B cells to produce these antibodies. But these antibodies are limited, right? They stay in your bloodstream. They don't cross the placenta, right? They aren't as harmful as some of the other antibodies. So for other antigens, they're T-dependent. A T cell has to help out that B cell. We want to make sure that we make that antibody, that we really need to make that antibody before we make it. Because they can be quite powerful, as you saw, all those protective outcomes. So IgG is your most abundant. This is the longest lived antibody. This is your antibody of memory. And remember, this is the one that interacts with the natural killer cell via that antibody-dependent cellular cytotoxicity. It can cross the placenta. This is the only one that does that. And this is the one that creates the problem for the Rh antigen found on the surface of your red blood cells for women. If you produce this antibody because you don't have the antigen and your baby does have the antigen, if this crosses the placenta, it will attack the baby's red blood cells. This is known as hemolytic disease of the newborn. And so it's one of those disorders that we'll talk about later when we get to disorders. It's also present in the colostrum. Anyone know what colostrum is? The first milk mom makes, right? It is a specialized milk, right? I think it's the first 24 hours or something like that. So even if you're not going to breastfeed, they usually try to get you to breastfeed in the hospital when you first deliver to give that particular milk to your baby. Because what your body does is it chocks it full of all the IgGs that you've produced over your lifetime. And so you pass that on to your baby. That's passive immunity. It's not long-lived. Eventually those antibodies will deteriorate and the baby won't have that protection anymore. But it gives them that level of protection until they can get vaccinated. Because they didn't make the antibodies, they don't have the memory B cells to make it again. But just giving them antibodies, those work too. And then, as I said, IgA, in your circulation, it's that monomer, but mostly we secrete this one in the dimer form. This is the most abundant antibody you make. You make gazillion tons of it a day for all your mucus, your tears, and then again, if you're breastfeeding, it's produced all the time in your breast milk to help protect your baby's digestive tract. And then there's IgE, as I said, not my favorite. Binds to mast cells and basophils, releases things like histamine, right? Causes inflammation and congestion against things that you don't necessarily need to be inflamed and congested over, like pollen, right? And dust mites, right? These things won't actually grow in your body, right? But for some of us, we respond quite aggressively to it as if it was something that would harm us. All right, so as I said, next time I'll back up. I don't think anybody has a really old edition of your textbook, but if you happen to, the table in the book had a really bad typo at one of the editions at one time, where it had these two pictures switched. So, be leery of that.
Have a good day.
You too? Let me click on